Yeah, yeah, yeah. On me, yeah. No escaping from my mind, but low key. You
And for our first question, which enzyme initiates DNA replication? A. DNA polymerase. B. RNA polymerase. C. Helicase. Or D. Primase. The correct answer is letter D. Primase. Primase initiates DNA replication by synthesizing a short RNA primer on the template strand. This primer provides a starting point for DNA polymerase to begin adding nucleotides. Next question. What unwinds the DNA double helix? A. Topoisomerase. B. Ligase. C. Helicase. Or D. Polymerase. The correct answer is letter C. Helicase. DNA helicase unwinds the double helix by breaking hydrogen bonds between the base pairs, creating the replication fork and allowing other enzymes to access the DNA strands for replication. Who discovered the Okazaki fragments? A. Reggie and Chuneko Okazaki. B. Reggie and Chewbacca Okazaki. C. Regine and Chupeko Okazaki. Or D. Raven and Chuneko Okazaki. The correct answer is letter A. Reggie and Chuneko Okazaki. Reiji Okazaki and Juneko Okazaki discovered Okazaki fragments in 1968. These are short DNA segments synthesized on the lagging strand during replication, later joined by DNA ligase to form a continuous strand. Next question. What is the direction of leading strand synthesis? A. 3' to 5', B. 5' to 3', C. 3' to 3', or D. 5' to 5'. And the correct answer is letter B. 5' to 3'. The leading strand is synthesized continuously in the 5' to 3' direction, following the same direction as the replication fork's movement. DNA polymerase adds nucleotides to the 3' end of the growing strand. Which enzyme seals gaps between Okazaki fragments? A. DNA ligase. B. DNA polymerase. C. RNA polymerase. Or D. Topoisomerase. And the correct answer is letter A. DNA ligase. DNA ligase seals gaps between Okazaki fragments by forming phosphodiester bonds, joining the sugar phosphate backbones to create a continuous DNA strand on the lagging strand. What enzyme corrects DNA replication errors? A. Reductase, B. DNA polymerase, C. RNA polymerase, or D. Topoisomerase. The correct answer is letter B. DNA polymerase. DNA polymerase corrects replication errors through its proofreading activity. It detects mismatched nucleotides, removes them using its 3' to 5' exonuclease activity, and replaces them with the correct nucleotide before continuing DNA synthesis. What forms at the replication fork? A. Leading strand, B. Lagging strand, C. Okazaki fragments, or D. Replication bubble. And the correct answer is letter D. Replication bubble. The replication fork is part of the replication bubble, where the DNA double helix is unwound and both the leading and the lagging strands are synthesized. Which structure stabilizes the replication fork? A. Replication complex, B. Helicase, C. Topoisomerase, or D. Single strand binding proteins. And the correct answer is letter D. Single strand binding proteins. Single strand binding proteins, or SSBs, stabilize the single stranded DNA at the replication fork by binding to the exposed strands, preventing them from reannealing or degrading while replication occurs. What are Okazaki fragments? A. Short DNA segments, B. Long DNA segments, C. RNA primers, or D. DNA polymerase. And the correct answer is letter A. Short DNA segments. Okazaki fragments are short, newly synthesized DNA segments formed on the lagging strand during DNA replication. Because the lagging strand is synthesized in the opposite direction of the replication fork, it is produced in small, discontinuous fragments. These fragments are later joined together by DNA ligase to form a continuous strand. And for our last question, which protein promotes DNA replication? A. Cyclin dependent kinase, B. Origin recognition complex, C. Helicase, or D. DNA polymerase. And the correct answer is letter C. Helicase. The DNA helicase protein promotes DNA replication by unwinding the DNA double helix ahead of the replication fork. This action creates two single stranded templates allowing other enzymes such as primase and DNA polymerase to synthesize new strands.